Hello friends, good afternoon and welcome to EduSite Live Lectures. Dear friends, today in food microbiology we will be talking about food-borne infections. To discuss this topic we have with us our subject expert Dr. Pavas Goswami. Dr. Goswami is Assistant Professor in Department of Microbiology in Bhaskaracharya College of Applied Sciences, University of Delhi. Without further ado, I would like to welcome Sir to our studios and request him to start the lecture. Welcome, Sir.
Thank you. Good afternoon, friends. Today I will be talking about food-borne infections. You know, general term food poisoning is used, but food poisoning include two types of diseases. One is food-borne intoxications, which we have discussed earlier, and another is food-borne infections. The difference between the two is in food-borne intoxication, preformed toxins are present in the food, while in food-borne infection, live cells are required and they enter the body through food and cause infection through various mechanisms. Today we will discuss food-borne infection caused by Escherichia coli, Salmonella and Shigella. We will also talk about general principles of food-borne infections. What are the things our body can do to inhibit such organisms and how these infecting agents overcome the resistance caused by our body and cause infection. We will also study various virulence factors, mechanisms of these diseases, how they infect and produce toxins inside the body and cause disease. We will also study the transmission of these diseases, what are the foods involved in these diseases, how we can diagnose these diseases and what are the possible treatments for these diseases. So, starting with food-borne infections. Food-borne illnesses are caused by the entrance of bacteria into the body through ingestion of contaminated food and the reaction of body to their presence or to their metabolism. Here is transmission of food-borne diseases. They are usually transmitted by fecal oral route. Fishes are having these microorganisms and through feces they can contaminate fingers or water. Fingers can contaminate food utensils plus toddler objects which are the fomites, they can be contaminated and these finally lead to mouth and they can enter the body. Similarly, water used for washing or preparation of foods can be used and can act as vehicle for these pathogens and finally it will enter the body. Insects can also act and carry these organisms to the body. Now, coming to the human digestive tract. Human digestive tract has tongue, epiglottis, esophagus, stomach, gallbladder, pancreas, then small intestine and large intestine. To cause disease, microorganism or pathogen should be able to cross the acidity barrier which is provided at the stomach end, then it should be able to grow at high concentration of bile etc. which are present in the intestine. It should compete with the organisms or normal flora of the body. So, these are the prerequisites which should be carried out for invasion of host. So, first is survival of the passage through the extremely acid environment of stomach. So, organism can survive this stomach acidity either by the protective effect of food, like organism is present in the food and food may act as matrix and it may save this organism and allow it to pass the acidity which is created at the stomach level. Now, this is important, and for passing through this kind of acidity, there is requirement of high number of organisms if organism do not have some other mechanisms. And another thing is adaptive acid tolerance mechanisms. If organism is having adaptive acid tolerance mechanisms, then it can tolerate acidity on its own and pass through stomach. Once it has passed through stomach, it should colonize and attach to the walls of digestive tract, which is specific for the specific organism. Some organisms might invade intestine, might attach to small intestine, some might attach to large intestine depending on the receptors present over there. Then it should possess ability to defend itself from host's immune response. If it is unable to tolerate host immune response, then this organism will be killed and it will not be able to cause disease. Then once it has reached digestive tract, it should multiply over there, and it should tolerate conditions like bile salts etc., which are present in the digestive tract. Some organisms have mechanisms such as enzymes, bile salt hydrolases, which cause detoxification of bile salts. Then another point, it should be able to compete with the normal microflora of the gut. If it is unable to compete, then it will not cause any disease, and it should also have virulence factors for causing disease. Now, these three members, E. coli, Shigella and Salmonella, these are the members of the family Enterobacteriaceae. This is a large group of bacteria, largest family of bacteria, and members of this family are often called enteric bacteria, as some of them are normal inhabitants of small and large intestine. They are also found in soil, water and decaying matter, and some of them are parasites on animals, while some are plant pathogens. Enterobacteriaceae are gram-negative straight rods, facultative anaerobes with simple nutritional requirements, and enterobacteria are divided into two groups based on their fermentation products. Either they will produce acidic products such as lactate, succinate, formate etc., or they might produce alkaline products such as butanediol, ethanol, carbon dioxide. The bacteria which we are discussing, that is E. coli, Salmonella and Shigella, are from mixed acid fermenter group. There are several other bacteria involved included in Enterobacteriaceae like Yersinia pestis, Salmonella we have discussed, then Klebsiella, Irvinia amylovora and Irvinia keratovora. These are basically plant pathogens. Now coming to our first organism, that is Escherichia coli or E. coli. These are gram-negative rods, facultatively anaerobic, normal flora of humans, and they are motile with peritrichous flagella. These are positive for methyl red reaction, that is they produce acid. They are human pathogens, cause UTI, diarrhea, and they are used as indicator for fecal contamination of water. This is important because E. coli is present in fishes, and if it is being found in water body, that means that this water body or water sample has been contaminated with fishes. So, large number of E. coli can be found and they are very easy to detect. This easy detection is helpful and is helps in use of these organisms as indicator organisms. These organisms are used as model organisms for research purposes also. Coming to taxonomy, their domain is bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Enterobacteriales, family we have discussed Enterobacteriaceae, and genus Escherichia. Coming to biochemicals, biochemical reactions, E. coli produces positive tests for indole, lysine, decarboxylase. Then mannitol fermentation produces gas from glucose and lactose. Typical colonial morphology is there and it produces metallic green sheen when grown on EMB agar. EMB agar is eosin methylene blue agar, and this agar is having peptone, lactose, sucrose, dipotassium phosphate, eosin Y, methylene blue, and the pH of this agar is 7.2. In this, eosin Y and methylene blue are indicators, and methylene blue also act as selective agents. These are the colonies of E. coli growing on EMB agar. You can clearly see the green sheen, green metallic sheen, which is being produced by these organisms. This is the diagram of E. coli cell. Here is the flagella, then capsule, LPS, lipopolysaccharide, cell membrane, and fimbriae. Now, these components of this cell are very important as far as virulence is considered. Capsule contains antigens, these are denoted by K, letter K, and are called K antigens, and these may play a role in attachment of this cell with the enterocytes, with other body cells. Then lipopolysaccharides are present, which again play a role in attachment, and bacteria are classified based on serogroups of O antigen and flagellar antigens. Coming to antigens, O antigens are the most external part of the cell wall, lipopolysaccharide, and consists of repeating units of polysaccharide. Same O specific polysaccharides contain unique sugars. O antigens are resistant to heat and alcohol, and they may be associated with specific human diseases. Specific O types of E. coli are found in diarrhea and urinary tract infections. K antigens are capsular antigens, these are polysaccharides, and K antigens may interfere with agglutination by O antisera. They may be associated with virulence, and E. coli strains producing K1 antigen are prominent in neonatal meningitis, and K antigens of E. coli cause attachment of the bacteria to epithelial cells prior to gastrointestinal or urinary tract invasion. Then H antigens are there. These H antigens are located on flagella, denatured or removed by alcohol and heat, and these antigens are determinants of the amino acid sequence in the flagellar protein. Coming to important part, diseases caused by E. coli. E. coli is responsible for causing diarrheal diseases which are of worldwide origin. There are several groups of E. coli which are involved in causing these diseases. The small or large bowel epithelial cells adherence properties are encoded by genes on plasmids, and these are important for causing diarrheal diseases. The toxins are also produced, and these are often plasmid or phage mediated. Diarrheogenic classes of E. coli are there. These classes are based on disease syndromes and characteristics, their effect on certain cell cultures and serological groupings. Diarrheogenic classes of E. coli include enteropathogenic E. coli, enterohemorrhagic E. coli, enterotoxigenic E. coli, enteroaggregative E. coli, enteroinvasive E. coli. So, these are different groups of E. coli that cause diarrhea. Their mechanism of causing diarrhea is different, though there are some similarities, as I discussed that there are some prerequisites for causing infection. So, these bacteria are having some similarities by which they enter the body and they cause disease, but mechanism may differ, and it is differing for these diseases. Common themes in E. coli virulence include colonization of mucosal site, evasion of host defenses, multiplication and host damage. This is the common factors for all the E. coli which cause diarrhea. Coming to the first group, enteropathogenic E. coli. Enteropathogenic E. coli is an important cause of infantile diarrhea, especially in developing countries. EPEC were previously associated with outbreaks of diarrhea in nurseries in developed countries. EPEC adhere to the mucosal cells of the small bowel. Chromosomally mediated factors are present which promote tight adherence of EPEC. There is loss of microvilli, formation of filamentous actin pedestals or cup-like structures, and occasionally entry of EPEC into the mucosal cells. Rarely entry of EPEC occurs. Normally EPEC do not enter the mucosal cells. Characteristic lesions are formed which can be seen in electron micrographs, and the result of EPEC infection is watery diarrhea, which is usually self-limited but can be chronic also. Enteropathogenic E. coli is an extracellular pathogen. As I discussed, it do not enter the cells, but it cause disease by binding to the surface of host cell and directly injecting virulence factors into the underlying cell through type 3 secretion system. These translocated proteins then interact with host cell components and alter signaling pathways, resulting in disease. During infection, EPEC induces a characteristic attaching and effacing AE histopathology on gut enterocytes. AE lesions are characterized by the localized effacement of microvilli and marked cytoskeletal changes, including the accumulation of polymerized actin directly beneath the adherent bacteria. The reorganization of actin forms pedestal-like structure upon which the bacterium resides. Here is the diagram. This is the bacterial cell EPEC. It produces ESPs, which are EPEC secreted proteins. They form a channel in the host cell, and through this channel translocated intimin receptor enters and binds to the membrane of the cell. Now, when this organism express intimin, it binds to TIR, and other signals are also sent which leads to actin aggregation, forming pedestal like structure. See here, this is the pedestal formed by the host cell, and this is the bacteria. Here is the diagram. Initially there is adherence by bundle forming pili, and this is followed by intimate attachment and actin condensation and microvillus effacement. EPEC diarrhea has been associated with multiple specific serotypes of E. coli, and strains are identified by O antigen and occasionally by H antigen typing. Infection generally causes acute diarrhea, but in severe cases it may lead to protracted diseases. Aside from profuse watery diarrhea, both vomiting and the development of fever are also observed in EPEC infection. Based on morbidity and mortality, EPEC strains remain a significant health threat to children worldwide. The duration of EPEC diarrhea can be shortened, and the chronic diarrhea can be cured by antibiotic treatment. Another group is that of enterohemorrhagic E. coli. EHEC are loosely defined by a combination of the symptoms they produce and the virulence factors they possess. The disease defining symptoms of EHEC is hemorrhagic colitis, that is, they cause bloody diarrhea. EHEC strains produces toxins which are Shiga-like toxins or Shiga toxin, also called as virotoxin, because it is toxic to viro cell lines, and these toxins, the genes for these toxins may have been transferred directly or indirectly from Shigella. The toxin is 70,000 Dalton protein composed of a single A subunit and 5 B subunits. B subunit is for binding and A is the active subunit. B subunit binds to the tissue, particularly to globotriacyl ceramide receptor, and A subunit inactivates the 28S ribosome, thus affecting and blocking the protein synthesis. Endothelial cells in GB3 receptors are primary target, and due to this HC hemolytic colitis and hemolytic uremic syndromes are caused. In HUS, hemolytic uremic syndrome, renal glomeruli are associated, and this toxin can also indirectly damage cells by causing other effects such as tumor necrosis factor release. The EAE chromosomal gene is ubiquitous among EHEC strains, encoding for an outer membrane protein which is associated with the attachment. Enterohemorrhagic E. coli mechanism is attachment to the cell and synthesis of Shiga toxin, thus inhibiting the protein synthesis and leading to cell death. Initial symptoms of hemorrhagic colitis generally occur in 1 to 2 days after eating the contaminated food. However, longer periods have also been reported. Symptoms start with mild non-bloody diarrhea and may be followed by a period of crampy abdominal pain and short-lived fever. The initial diarrhea increases in intensity during the next 24 to 48 hours to a 4 to 10 day phase of overtly bloody diarrhea accompanied by severe abdominal pain and moderate dehydration. Hemolytic uremic syndrome is another complication caused by enterohemorrhagic E. coli, and its onset is approximately a week after the onset of gastrointestinal symptoms. Characteristic symptoms of HUS are pallor, intravascular destruction of red blood cells, that is microangiopathic hemolytic anemia, depressed platelet counts, thrombocytopenia, lack of urine formation, swelling, edema, and acute renal failure. Hemolytic uremic syndrome occurs most often in children under the age of 10. Other HUS associated complications may include seizures, coma, stroke, colonic perforation, pancreatitis, and hypertension, and approximately 15% of cases lead to early development of chronic kidney failure. Insulin dependent diabetes may also persist in HUS. These are various strains responsible for HUS. In this, most important is E. coli O157H7. Its serotype O is 157 type and H is 7 type, and these are other strains which may be involved in hemolytic uremic syndrome. Foods that are involved include undercooked ground beef, raw milk, unpasteurized apple juice, dried cured salami, lettuce, produce from manure fertilized garden, radish sprouts, alfalfa sprouts, yogurt, sandwiches, water. Reservoirs and sources of E. coli are cattle. Young animals tend to carry E. coli O157H7 more frequently than adults because normal microflora is not set and it does not provide much competition to this E. coli strain. E. coli O157H7 levels in calf fishes range from around 10 raised to power 2 to 10 raised to power 5 CFU per gram, and more than one strain of E. coli O157H7 can be isolated from fishes of the same animal or from different animals within the same herd. Deer, sheep are also carriers for E. coli O157H7, and water which is contaminated by cattle feces or feces from other organisms or humans may also act as a vehicle for E. coli O157H7. Now, what are the precautions to prevent E. coli O157H7 infection? These include thoroughly heating of food before eating, drinking of only pasteurized milk and juice, washing of fresh fruits and vegetables before eating, washing of hands thoroughly after handling animals, washing hands thoroughly after changing diapers, preventing use of fresh manure from ruminants to fertilize vegetables or fruits, and avoid swimming in lakes or ponds used by cattle, and drinking surface water that has not been properly treated. Now, another group of E. coli is enterotoxigenic E. coli. This is a common cause of travelers diarrhea, and it produces colonization factor antigens which are specific for humans that promote adherence of ETEC to epithelial cells of the small bowel. The ETEC disease syndromes are rarely accompanied by fever, and the diarrhea is sudden. Estimated amounts of 10 raised to power 8 to 10 raised to power 10 CFU are necessary for causing diarrhea in adult humans. These ETEC strains are known to produce toxins. Two types of toxins are produced, a labile toxin and a heat stable toxin. Labile toxin is of molecular weight of 80,000, and this toxin is inactivated at 60 degrees centigrade in 30 minutes. It is having subunit B which attaches to GM1 ganglioside at the brush border of epithelial cells and facilitates the entry of subunit A which activates enzyme adenylyl cyclase. The A subunit of this toxin cause ADP ribosylation of GTP binding protein and stimulates adenylate cyclase activity. ADP ribosylation results in increased concentration of intracellular cyclic AMP, and cyclic AMP dependent protein kinase is activated, leading to supranormal phosphorylation of chloride channels located in the apical epithelial cell membranes. This increases the concentration of CMP and also causes the outflow of chloride ions, plus inflow of sodium chloride is also inhibited, and this results in diarrhea. Thank you. Another toxin of E. coli enterotoxigenic strain is stable toxin, and this heat stable toxin is having molecular weight of somewhere 1500 to 4000, and this is under the genetic control of heterogeneous group of plasmids. This is a heat stable toxin which can withstand 100 degrees centigrade for 15 minutes. This toxin activates guanylyl cyclase and increase the concentration of cyclic AMP. Some strains produce both the toxin. The stable toxin is 18 or 19 amino acid peptide with a molecular mass of 2 kilodalton. Toxin is released by diffusion across the outer membrane, and the receptor guanylate cyclase C binds to it. Once this binds to this enzyme, this pump, it increases the concentration of cyclic GMP, which again have the same effect as labile toxin. it causes secretion of chloride ions, while absorption of NaCl is hampered. Enterotoxigenic E. coli carries a plasmid on which genes for LT and ST are present, and there is no definite association of enterotoxigenic E. coli with EPEC strains. Colonization factors are also present, which are responsible for colonization of ETEC. Then another important group is enteroinvasive E. coli. Enteroinvasive E. coli produce diseases by invading intestinal mucosal epithelial cells. EIEC strains are biochemically, genetically and pathogenetically related to Shigella species. The invasion related plasmid has been designated P-INV. Their pathogenesis involve epithelial cell penetration, lysis of the endocytic vacuole, intracellular multiplication, directional movement through the cytoplasm and extension into adjacent epithelial cells. This is the mode of action of enteroinvasive E. coli. You can see here, this is the pathogen. It enters the cell and move out of the phagosomal vesicle and spread through other cells laterally, causing disease. Then enteroaggregative E. coli. This E. coli is characterized by their characteristic pattern of adherence to human cells. Stacked brick pattern is produced by this E. coli, and it may also produce ST-like toxin and hemolysin. Here these E. coli cells aggregate like stack of brick and they deliver cytotoxin causing the disease. Coming to the treatment, no single specific therapy is available. Sulfonamides, ampicillin, cephalosporins, aminoglycosides have been used, but variation in susceptibility is great and laboratory tests for antibiotic sensitivity should be carried out. Otherwise, it may lead to the problem of antimicrobial resistance. Treatment of traveler's diarrhea can be carried out by giving bismuth subsalicylate suspension. This suspension is known to inactivate the toxin produced by E. coli cells, and regular doses of tetracyclines or other antimicrobial drugs may also be given. But it is better to take precaution to live in a sanitized environment, and antibiotic treatment may be given briefly such as ciprofloxacin, trimethoprim may be given as prophylaxis. Coming to epidemiology, the enteric bacteria are present in normal intestinal tract. They are found in water or milk, and it is not easy to control the normal flora of the body. Sanitary measures should be taken to control these organisms. Infected poultry meats and eggs must be thoroughly cooked, and carriers must not be allowed to work as food handlers. Now, coming to the next disease, that is shigellosis. This disease is caused by Shigella, which is gram negative, non-spore forming, non-motile, facultative anaerobes, do not ferment lactose. These cells are oxidase negative, produce acid only from sugars. They do not grow on citrate as sole carbon source and do not produce H2S. These are the causative agents of bacillary dysentery, and some of the important species include Shigella dysenteriae, flexneri, boydii and sonii. Their temperature growth range is 10 to 48 degrees centigrade and pH range is 5 to 8. However, best growth occurs in a pH range of 6 to 8. Very few cells of Shigella, only say 10 to 100 cells are required to cause disease. Shigella infection is limited to the gastrointestinal tract and blood invasion is quite rare. bacterial cells are taken up by M cells and are transported beneath the epithelium, where these cells are taken up by macrophages, and Shigella cells induce apoptosis in macrophages and free bacteria invade epithelial cells. This is the diagram of M cell. It is having a basolateral pocket in which macrophages and other immune cells are present. Shigella is taken up, it enters through M cell and is then ingested by macrophages. Killing of macrophages leads to inflammation and there is movement of NK cells and PMNLs towards the site of infection. PMNLs disintegrate epithelial lining and this results in tissue destruction, which facilitates the invasion of more bacteria. And finally, PMNLs kill bacteria. So, this is the diagram. Shigella enters through M cell, it is taken up by macrophages, it causes apoptosis of macrophages and this leads to inflammation. Shigella then infects through the basolateral surface and it moves and causes infection. Meanwhile, PMNLs and natural killer cells are also activated. Here cell destruction takes place followed by repair and finally these cells PMNLs etc. they kill Shigella and control the infection. Endotoxins produced by Shigella, these include lipopolysaccharide and exotoxin again is AB type of toxin which inactivates 60S ribosomal unit. It is same like EHEC toxin. Symptoms takes place develop in 1 to 7 days, but usually takes less than 4 days. There are abdominal cramps, fever, chills, diarrhea, watery stools containing blood, mucus and pus. There is tenesmus, headache, prostration, nausea and dehydrations. Foods involved are milk, beans, potato, shrimp, turkey, salads and apple cider. Control measures include practicing of personal hygiene, chilling of foods rapidly, preparations of foods in a sanitary manner, cooking of food thoroughly, disposing of sewage in a sanitary manner and control of flies. Diagnosis is by growing these cells on XLD agar, xylose lysine desoxycholate agar and confirming the presence of Shigella by carrying out TSI test, triple sugar iron test, in which alkaline slant and acidic butt are produced. There is no production of H2S, no indole formation and no motility. Antibiotics such as trimethoprim, sulfamethoxazole, ciprofloxacin may be given for treatment of this disease. Next disease we will talk about is salmonellosis. It is caused by Salmonella, which are often pathogenic for humans and animals when they are acquired by oral route. They are transmitted from animals and animal products where they cause enteritis, systemic infection and enteric fever. Salmonella grow rapidly on simple media, but they almost never ferment lactose or sucrose. They form acid and sometimes gas from glucose and mannose. They are positive for H2S production and they survive freezing in water for long periods. Salmonella are resistant to certain chemicals such as brilliant green, sodium tetrathionate, sodium deoxycholate that inhibit other enteric bacteria. This fact can be used for synthesizing the media for isolation of Salmonella. The classification of Salmonella is complex because the organisms are a continuum rather than a defined species. The members of genus Salmonella were originally classified on the basis of epidemiology, host range and biochemical reactions. The names Salmonella typhi, where we write genus and species both in italics, is not correct when we use it for Salmonella. In this, the correct way of writing is Salmonella, genus name in italics, and Typhimurium as starting with a capital letter and which is not in italics, normal font, because this organism is this Typhimurium is a serotype. Most of the Salmonella which cause disease are included in DNA hybridization group 1. Salmonella infecting humans are included in DNA hybridization group 1, and these are usually considered as Salmonella enterica subspecies enterica, and all these other other names Typhimurium, Paratyphi, etc. are serotypes. Variation is there in antigens of Salmonella. They lose H antigen, that is flagellar antigen, then loss of O antigen may also take place, and VI, that is capsular antigen, may also be affected. Antigen may be acquired or lost in the process of transduction. Coming to pathogenesis and clinical findings, the organisms almost always enter via the oral route, usually with contaminated food or drink, and the infective dose in humans is 10 raised to power 5 to 10 raised to power 8 Salmonella. Among the host factors that contribute to resistance of Salmonella infection are gastric acidity, normal intestine microbial flora, and local intestinal immunity. Coming to typhoid fevers, this syndrome is produced by only a few of Salmonella. Example is that of Salmonella typhi. The ingested Salmonella reach the small intestine, from where they enter the lymphatics and then the bloodstream. They are carried by the blood to many organs including intestine, and these organisms multiply in intestinal lymphoid tissue and are excreted in feces. After an incubation period of 10 to 14 days, there is fever, malaise, headache, constipation, bradycardia, and myalgia. The fever rises to a high plateau, spleen and liver become enlarged, rose spots develop usually on skin of abdomen or chest. Then white blood cell count is normal or low, and they may cause intestinal hemorrhage and perforation and high mortality rate of 10 to 15%. Treatment with antibiotics has reduced the mortality rate to less than 1%. The principal lesions caused by Salmonella are hyperplasia and necrosis of lymphoid tissue, hepatitis, focal necrosis of the liver, and inflammation of gallbladder, periosteum, lungs, and other organs. Another important disease is enterocolitis. This is the most common manifestation of Salmonella infection. In United States, Salmonella typhimurium and Salmonella enteritis are prominent, but enterocolitis can be caused by any of more than 1400 group 1 serotypes of Salmonella. 8 to 48 hours are required for causing disease, and after 48 hours of ingestion there is nausea, headache, vomiting, profuse diarrhea, and low grade fever, which usually resolves in 2 to 3 days. These symptoms are usually accompanied by prostration, muscular weakness, faintness, moderate fever, restlessness, drowsiness, watery greenish foul smelling stools, and twitching. Inflammatory lesions of the small and large intestine are present. Bacteremia is rare except in immunodeficient persons. Blood cultures are usually negative, but stool cultures are positive for Salmonella and may remain positive for several weeks after clinical recovery. Here is the example of incidence and vehicle food. An incidence of Salmonella enteritidis was observed in 2,24,000 persons, and the vehicle food was ice cream which was produced from milk, but was transported in tanker trucks that had hauled liquid eggs earlier. So, Salmonella from eggs have contaminated the ice cream. In another outbreak, 2% milk produced by a single dairy plant was responsible for around 2 lakh persons cases. Then as Newport, 3,400 persons were involved, where potato salad prepared and stored for up to 16 hours at improper holding temperature was responsible for the outbreak. Prevention and control of Salmonella can be carried out by avoidance of contamination, destruction of the organism, and prevention of growth of Salmonella. Avoidance of contamination of food with Salmonella from sources such as diseased human being and animals and ingredients is required. Contaminated eggs may carry Salmonella and may lead to contamination of other foods. Then destruction of organism can be carried out by heating. Salmonella Septemberg is required to heat at 54.4 degrees centigrade and in liquid whole egg, and this strain is most heat resistant, and this was the temperature used, and this also leads to destruction of egg alpha amylase. So, it has been suggested that alpha amylase test may be used as means of determining the adequacy of heat pasteurization of liquid egg. In milk, the criteria is enzyme alkaline phosphatase, which is inactivated by pasteurization of milk. Prevention of growth of Salmonella in foods by adequate refrigeration or other means can be carried out. Care and cleanliness in food handling and preparation may be used. Food handlers which are carriers of Salmonella should not be used. Rats and other vermin insects should be prevented. Then ingredients should be inspected for Salmonella. Inspection of animals and meat at packing should be done, and food should not be kept at room temperature. Competitive exclusion can also be carried out to reduce Salmonella carriage in poultry. In this, fishes from healthy birds which are free of Salmonella is given to young chicks. This leads to establishment of microflora of young chicks, and Salmonella cannot compete with this microflora, and disease is prevented by its exclusion. Similarly, older birds which are having microflora can be administered with antibacterial agents, and again they can be administered with competitive microbial biota, which will lead to exclusion of Salmonella. And the principle behind this is competitive exclusion of Salmonella by the native gut microbiota. Diagnosis of Salmonella can be carried out by using certain serological tests, by isolating of organisms, or by looking for presence of antibodies against Salmonella. Blood for culture may be taken repeatedly in diagnosis of Salmonella for enteric fevers and septicemias. Then stool specimens may also be taken for enteric fevers and enterocolitis. Positive culture of duodenal drainage establish the presence of Salmonella in the biliary tract carriers. Differential medium cultures can be used, and these permits rapid detection of lactose non-fermenters, McConkey deoxycholate medium, and here those organisms which are not fermenting lactose will give colorless colonies. Gram positive organisms are inhibited on these medium. Bismuth sulfide medium permits rapid detection of Salmonella where black colonies are formed due to H2S production. Selective medium cultures for Salmonella include Salmonella shigella agar, Hecton enteric agar, xylose lysine deoxycholate agar and deoxycholate citrate agar. Enrichment cultures can be carried out. From this specimen we can use selenite F or tetrathionate broth, and this broth does not inhibit Salmonella, but it will inhibit other intestinal bacteria, and Salmonella can be enriched in this manner. Final identification is carried out by biochemical reaction patterns and slide agglutination tests with specific sera. Serological techniques include agglutination test. In this, known sera and unknown culture are mixed on slide. If clumping occurs, that means Salmonella is present, because the antibodies are used in these cases are specific for Salmonella, and it will give agglutination only with Salmonella and not with other bacteria. This test is particularly useful for rapid preliminary identification of cultures. Commercial kits are also available depending on the serogroups of O antigens. Then serologic techniques can be used. These include tube dilution agglutination test, and in this antibodies produced against Salmonella O and H antigens are detected. Serial dilutions of unknown sera is carried out, and then these dilutions are used for agglutination tests. The interpretive criteria is when single serum specimens are tested vary, but a titer against O antigen of greater than 1:320 and against the H antigen 1:640 is considered positive. High titer of antibody to VI antigen also occur in some carriers. Results of serological tests of Salmonella infection must be interpreted cautiously, because the possible presence of cross-reactive antibodies may limit the use of serology. Treatment of Salmonella is by antimicrobial therapy, in which ampicillin, trimethoprim, sulfamethoxazole and cephalosporin is used. Multiple drug resistance is transmitted genetically by plasmids, and so susceptibility testing should be done to select a proper antibiotic. Coming to epidemiology, the feces of persons who have unsuspected subclinical disease or are carriers are more important source of contamination. Many animals including cattle, rodents, fowl are naturally infected, and they have bacteria in their tissue, excreta or eggs. High incidence of Salmonella is observed in commercially prepared chickens. The incidence of typhoid fever has decreased, but the incidence of other Salmonella infections have increased in United States. The problem is aggravated by widespread use of animal feeds containing antimicrobial drugs, which leads to selection of drug resistant Salmonella and their potential transmission to humans. Carriers are present which carry Salmonella. They might not be having the disease, but they carry Salmonella in their body, and after subclinical infection, some individuals may continue to harbor Salmonella in their tissues for variable lengths of time. 3% of survivors of typhoid become permanent carriers, harboring the organisms in the gallbladder, biliary tract, or rarely in intestine or urinary tract. The sources of infection of Salmonella are contaminated food and drink. Water is an important source of contamination. Contamination of water with feces results in epidemics. Milk and other dairy products are also important. Their contamination with fishes results in outbreaks. Inadequate pasteurization and improper handling of milk products is also responsible for outbreaks, and shellfish contamination from water may also lead to outbreaks. Dried or frozen eggs may also act as carrier for Salmonella. Vehicle food for Salmonella. Meat and meat products from infected animals are also responsible. Animal dyes such as carmine used in foods and cosmetics are also responsible for Salmonella infections. Then household pets such as turtles, dogs, cats may also be responsible. Now coming to prevention and control for almost all foodborne pathogens including Salmonella, sanitary measures must be taken to prevent contamination of food. Infected food, meats and eggs must be thoroughly cooked, and carriers must not be allowed to work as food handlers, and they should observe strict hygienic precautions. These are some of the references which have been used for preparation of this presentation. Thank you.
On that note, I would like to thank sir for this very enriching discussion, and thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.